All right, welcome everyone. I'm excited to start off this session. So today I'm going to be talking, and I'm actually really excited. This says genomics session, but it's all bird talks all the time, so let's go. I will be talking about a female-specific growth gene and the role that that could play in reverse sexual size dimorphism that we see in birds. So first, let me figure out how the clicker works. First, let's talk about the mystery of reverse sexual size dimorphism. So as you likely know, very few birds actually have larger females. So what could make these females at this tail end of the distribution different? So there have been multiple forces proposed to select for this. First of all, natural selection, such as niche or role partitioning, or reducing sort of the percentage change in body weight during the period when females are about to lay eggs, especially if they rely on quite acrobatic hunting in order to provision themselves. Also, sexual selection can play a role, such as in dunlins. There have been studies that show that the females consistently select the smaller males, which have more acrobatic flight displays. But we do not know what the underlying biological mechanism behind these could be, so I want to discuss that here. The biological basis of growth overall is controlled by many regulators, but two of the most important ones are aptly named the growth hormone as well as the growth hormone receptor. So when the ligand binds to this receptor, that signals growth both locally as well as throughout the body, in many tissues, creating this vertical growth, muscle mass growth, etc. You need to have the right amount of signaling, so if you don't get enough, this is a dwarf-rooster phenotype that has a broken growth hormone receptor gene. But if you get too much, you get pituitary gigantism, such as this famous man. But I want to know if changes in these genes could be related to sex-specific patterns of growth that we see in birds. So we were searching for growth hormone receptor duplication because we had previously explored a passerin-specific growth hormone gene duplication. And we do know that in other vertebrates, this gene has often been synchronously duplicated, sometimes not, with the receptor. So we're looking into that. Was that the case? No, that's not this talk, but I did find two distinct copies of this receptor gene in other avian taxa, and intriguingly, many of these taxa are known for their RSD. So it's actually a female-specific gene, as it is on the W chromosome. So growth hormone receptor, the main one that all birds have, is on the Z. So males actually have two copies and females have one. And similar to many other Z-linked genes, males do not dosage compensate across the chromosome, and they're actually, there's plenty of data showing that they express this gene more. So females have generally a disadvantage here. But we found that a few species actually do have a W copy retained. And this copy is divergent in coding sequence from the Z copy. So where did this W copy come from? We know that the receptor gene is ancestral on the autosome that was later recruited as the sex chromosome pair in birds. One example of this would be autosome 3 pair in alligators is homologous to the bird sex chromosomes. You can see here's the growth hormone receptor gene among a few other major markers. This was then recruited as the sex chromosome, with this becoming the sex-determining gene and being lost from the W chromosome, with, for example, the emu still keeping this type of karyotype. However, the vast majority of birds have highly degraded W chromosomes. There's also some other funky sex chromosome stuff happening in other birds that we'll hear about in later talks. Looking forward to it. Anyway, as this W is degrading, GHR is just one of the many genes that is most often lost from this chromosome, leaving female birds with only one copy. So I wanted to look at the gene tree, built one with this growth hormone receptor gene. And paleonaths, the earliest branching group, ostriches, et cetera, the weird guys, they tend to have a more complete W. And therefore, when we look at these gene sequences, their Z and W gene tend to cluster together, being most related to each other. Either it's still in a recombining region, or it's in a region that was more recently recombining. That's different from neonaths, all the other birds, where when we do see retention of W, it's actually clustering in its own clade, separate from the Z So these are no longer in a recombining region, and they are evolving on separate trajectories. So this retention of the W in these neonath birds is pretty rare among these genes that have left this recombining region. You can see these birds here are neonath examples that have kept their W. These are ones that have not. And you see as a percentage of W genes that they have retained, they're not very exceptional. So it's not just that their Ws are less degraded, they're actually specifically keeping this gene intact, which is really surprising considering that this gene is very, very large. And also it's in a region of the W that's filled with TE content, repeats, and other forms of degradation. So I wanted to then map the phylogenetic distribution of this GHRW copy. So sort of one representative per order here, and the yellow circles represent losses. So these are orders where we did not find any evidence. The blue is when we have good evidence or pretty much like full sequences, and green are partial sequences where either the quality of the genome assembly in these regions is not high enough to determine if there is a full gene, or it could be a biological reality that like only part of the gene remains. It's on its way to degradation possibly. But the takeaway here is that many of these clades where GHRW is present are noted for their RSD, such as these birds of prey. For example, you can tell this one on the right is the female owl, and that's not even among the most extreme examples in this group. You have literally birds of prey where the female is twice as large. But this is the most numerous number of species that we have with this W copy. So looking across one bird example kind of per order where we have RSD, as well as one bird example per order where we have GHRW, what does the overlap look like? So the majority of birds with RSD have GHRW, and vice versa. There are some that don't have one or the other. Some of the low quality genomes, not sure yet. But anyway, there is a concordance that is not confined to just one clade. But we wanted to look at if this correlation exists broadly. So before correcting for phylogeny, you can see here when the larger sex is female over here, there's a much higher percentage of species with GHRW. When you roughly correct by subsampling about one species per family, so removing like the 70 hawks and eagles that I had in the data set, you can still see that there is a higher percentage in there, but we do want to check that those are not just like one clade that's related to each other. We want to see if this is potentially a driver of this trait across the avian tree. So what we did was check for correlation after correcting for phylogeny. So we were able to do this because of the many repeated losses, means that there's sort of many separate retentions, something testable with correction for phylogeny. We actually constructed this tree using a subset of species for which many other scientists have gathered data on the sex-specific masses, and that were also present on this robust family-level tree that is constructed from UCE and Intron data. The details of the tree are not important. Please don't go blind trying to read it. But the important thing here is that we did find GHRW presence is categorically correlated with RSD. And I try to sort of point out several clades where it's bluer, and you see these kind of separate instances of larger females with RSD. Going on to the evidence that both of these gametologs or copies are actually expressed, we wanted to check on that, right? So it's correlated, but is it actually being used? Is it being expressed? We already knew that it had an intact intron exon structure and open reading frame. That's very important preliminary information, but is it being used? So we aligned RNA-seq reads from a California condor genomic and transcriptomic data set. So there was a female from whom the genome was assembled, and then her and another female were pooled together for a transcriptome. And in realigning these reads, we looked at the reads that were mapping uniquely to each one of these gametologs, and GHRW has 47% unique reads and 93% in GHRZ, and these reads map across the entire CDS, so we know that it's not just like one region driving this. So very exciting, and this higher number of unique reads in GHRZ is probably due to the overall much higher expression of this, which was estimated to be about 10% or 10 times the expression of the W. So this is fibroblast tissue. This was the only tissue available in which this was predicted to be expressed. The most interesting tissue to look at would be liver, but we don't have that yet, so more remains to be explored for sure. Lastly, I do want to talk a bit about the protein. So the GHRW protein, even though there is some divergence, most of that divergence is just synonymous changes in the coding sequence, and then many of the mutations are in the very large introns of this large gene. But there are two fixed differences in the amino acid sequence that contributed to the fact that you see those two separate neonaf clades. And these two fixed differences both occur in the same region of this protein. And they are, I know it looks a little loosey-goosey in this area of the structure, but both these sites are actually evolutionarily conserved, with, in particular, this site 335, which is also a glutamine in humans. When you mutate that, you get a pretty profound version of dwarfism. So the question is that affecting these birds? I mean, California condors are clearly not suffering from dwarfism. So more remains to be explored there, but it's very interesting that you see these fixed differences. So just to wrap up here, the takeaways are that the avian ancestor had two copies of growth hormone receptor, one on Z, one on W, but the vast majority of birds have lost their W copy during this degradation of the W that's quite extensive. However, in certain clades, even with quite degraded Ws, they are actually still expressing it, at least, for example, in the condor, and it has very few fixed differences from GHRZ, but these differences could be important for function. And we know that GHRW is retained in some lineages, especially in these iconic birds of prey with larger females, such as hawks, eagles, and owls. But it's not just confined to that taxon. It's also correlated to RSD across avian taxa. And overall, I just think this should be investigated further as a mechanism underpinning larger female body size across birds. So thank you all for your attention. I'd like to thank my 3 undergrad assistants for helping construct a database of avian sequences, and to my labs. So thank you. Yeah.
Asking as someone who doesn't do a lot of gene expression studies, could you explain the significance of the 47% unique reads and 93% unique reads?
Yeah, so these two genes have many regions of similarity. And so we wanted to make sure there were unique reads mapping to each of these genes. And GHR, essentially how the aligner works is when there is a read that aligns to both equally well, so a region that matches, it will just 50-50 flip a coin essentially and assign it to one of them. That was the aligner and the settings we were using. So if, because there is a much higher percentage of non-unique reads, that represents the fact that there's just a lower overall expression level. Fewer reads overall, because a much higher percentage of the reads that were mapped there were ones that were kind of like flip a coin 50-50. So a few of those non-unique reads could have been more appropriately mapped to the Z, but it's the unique ones we're really excited about because those represent the fact that it is actually, at least at a low level, being expressed.
Okay, hi, my name's Leonardo Campagna, and today I want to talk about the role of structural variation in shaping both coloration phenotypes and species differences in this radiation of seed eaters known as the cappuccino seed eater radiation. And so the idea behind this work is that if we can find recent speciation events and sort of catch speciation still in motion, we can leverage these to understand how species form. And so you'd find a situation like this middle Manhattan plot here, where if you have a system that's in the early stages of divergence, still has a lot of gene flow, you'd be able to look at the genome, compare species, and find those sort of isolated divergence peaks, and then ask, okay, what type of genes do we find in here? What genotypes do they mediate? And can we tie this to the origin of species? Conversely, or on the other hand, if you come in later in the game, find a situation like this where there's deeper divergence, more genome-wide divergence, it's very hard to sort of find the differences that were important in the beginning. And so the recent radiation we work on is known as the cappuccino seed eater radiation. These are males from the 10 different species. And you see that they can vary in their plumage patterns in a few different patches and a few different colors. So you have cap patches, throat, back, belly, and a few different tones of cinnamon and brown. And so we can use these coloration patterns from adult males to identify them in the field, and we can also identify them by their unique song. In contrast, females, juveniles, juvenile females all look the same. We cannot identify them morphologically. We identify them genetically now. So a little bit of background about the radiation. It's a continental radiation. You can find the birds there. I've overlaid the ranges. So a lot of birds in northern Argentina, southern Brazil, Paraguay, Uruguay. You'll see that area, that darker area, that's Esteros de Liberá in Corrientes, where we can find up to 7 species breeding in the same grassland. So they're, in general, highly sympatric, but there's areas where we find a lot more. And we've dated the radiation by dating the age of the node that the core radiation shares with the sister species to about 1 million years before present. And so I was saying they're highly sympatric, and they're found in this sort of structurally simple habitat because you wouldn't find them segregated into these different areas that you can see here, but you'd find them mostly in the grasslands back there, drier, a little bit elevated, sometimes a little bit inundated grasslands, and you find them there feeding and also reproducing. And so for a long time, we were using markers that sample the genome really sparsely, even reduced representation genomic techniques, and we always obtained the same result. We could tell apart the first 3 species, and then we kind of find that polytomy where the core radiation sits. So it's only when we first obtained whole genome sequences that we could see that individuals here were clustering together, individuals from the different species were clustering together. They show very little morphological differentiation, so if we didn't have the plumage patterns, we could not really identify them by beak or size or overall shape. We think this is because they're ecologically pretty similar. We are able to generate hybrids in aviaries, in controlled crosses, and we have a hard time identifying hybrids in the field. It is possible, but mostly because it can get confounded with different stages of plumage maturation. So there's a lot of sort of molting stages, intermediate individuals, and it's hard to have a precise ID. And so here I'm just showing you an example of a Manhattan plot between a pair of species. And so those differences that we were picking up in that genome-wide PCA are mostly localized to these divergence peaks, to a few divergence peaks that we find across the genome. And so you'll see that the average FST is very low, particularly for this comparison and across all the different pairwise comparisons that we can do. And these few peaks tend to contain genes that are involved in pigmentation, so genes that are well known to regulate melanogenesis, like TERP1 over there, OCA2 and HERC2. And we have a third sort of mystery peak there with a gene that we can't really connect easily to phenotype. So particularly this case is really cool because of work here led by Sheila Turbeck, where she found that despite sort of these genetic similarities, these 2 species are breeding in syntypy. So you can see they're color-coded or where she found the nests, and you'll see the scale, that's 500 meters. They also breed synchronously, so these nests are found sort of at the exact same time. The overlap there is shown in purple. But despite this, they mate assortatively. And so the females, which we can't identify in the field, are grouping together with the male of their correct species. So we're not seeing these mismatches. We can analyze these genome-wide data in a different way. In this case, this is work led by Cecilia Stalles. And what she did here is she took the pigment concentration of the feathers from different patches and used that as a phenotype in a GWAS. And you'll see that here she's comparing, say, a cinnamon throat with a black throat, and she'll associate that change to 2 of the melanogenesis genes that I've shown you before. Now she compares cinnamon to white. she gets another hit, in this case, a different gene involved in the melanogenesis pathway. And so we can do this strategy across different plumage patches and also do the FST outlier strategy across the different pairwise comparison. And we find all these candidate genes from the melanogenesis pathway. So we think this pathway has been tweaked. All upstream regulators, enzymes, transcription factors, transporters. It's been tweaked in different ways to generate the plumage divergence that we see in the group. So here I'm just zooming into a couple of peaks. This is the Agouti signaling protein peak. These are the two genes I've been talking about. And I'm overlaying the different pairwise comparisons. And you'll see that these peaks are coming up in different comparisons across independent species sometimes. And so this could be because you could have species-specific variants in some of these regions, or there could be shared variants by certain groups of species. So we can look at that in greater detail by looking at the genotypes in these regions. And you'll see up there, so the species with the black throat and black belly has that predominantly red haplotype, derived haplotype there, whereas the species that only have black throats have a slightly different one with a blue sort of area in the middle. And then the rest share the sort of ancestral variant that's mostly blue. In this case, we have the species with the white throat that mostly associates to that red haplotype. And so we can look at this in a different way, and I think it's a more efficient way, by trying to pull a topology from the ancestral recombination graph. And so here you'll see that most of the species share that ancestral variant, and then you have two derived variants, one for the black species, that one's species specific, and then the black-throated species share a variant in this region. And actually, having species-specific variants is the exception. Most of the cases across these peaks, we see shared variants across species. Just to show you another example here, that purple area is shared by those three species. And so as we move across the different peaks, we see shared variants in sort of unique combinations of species. Finally, I'll zoom in a little bit more to just make the final point that most of the divergent SNPs here that we find that show the highest level of differentiation, actually of all of them in these cases, are non-coding. So we think that these are presumably regulatory differences, and we're not really modifying the genes themselves. So one thing we had tried to get at about 7, 8 years ago was this question of, are these divergent areas associated with structural variants in any way? And so what we had done initially is we've obtained long reads for, in this case, 2 individuals of 2 species, tried to call the structural variants that we could, and in this case found that in general, inversions, deletions, insertions were not associated to our divergence peaks. There's some small ones over there. Most of them don't contain genes. There was one that was within an intron of a gene. So I wanted to revisit this question. And if you want to hear this in more detail, you should come to Maria's talk tomorrow here. She'll talk about how we built the pangenome, this pangenome for cappuccinos, to try to assess this landscape of structural variation. And today I'll just give you a little preview. So the idea here is that if you're working with a single reference, for example, and you're trying to map an individual that has an insertion and that's not present in the reference, you're going to lose those reads. You won't be able to genotype that insertion. And also, if you have short reads, you tend to have this effect when you have repetitive regions that they all pile up, and you're inferring the wrong sequence. And you need that red long read to span this region and sort of infer the accurate sequence. And so to do this, we obtained 32 haploid genomes from 7 species with PacBio Hi-Fi reads. We built this pangenome that here is sort of represented in graph form to try to capture as much as variation as possible, SNPs and all these different structural variants. And we mapped the short read data back to the pangenome. We used that as the reference to genotype our individuals. And so here I'm showing you an example of the graph from within a divergence peak. And you can see this is a very collinear area. You'll see SNPs represented there up in that region. That's, for example, a small T insertion. But then, if we zoom out, we have these regions as kind of blow up. So these are sort of complex rearrangements that have inversions, insertions, duplications, and they occur throughout the genome in different places. And so our question is, do these associate at all with the species differences or with these coloration areas responsible for shaping coloration? And to do this, we did both the GWAS and the FST outlier strategy. I'm going to show you the example for the eumelanin content of the belly, but the same message is true for other patches and with the FST outlier strategy. And so here we're using SNPs. You can see we identify 3 main peaks. I'll focus on those 3 peaks. The middle one is the agouti signaling protein peak. And we can do this again, but only with SVs. And so in this case, you'll see that with SVs, we mostly recover the first 2 peaks. But importantly, in orange are the true SVs, so those that are larger than 50 base pairs. And you'll see that none of them are above our threshold of significance. And so the take-home message here is that we can replicate the signal of the SNPs, but mostly with smaller indels. We don't really associate the bigger variants to these outlier regions. And we can identify quite a few of them, but they don't seem to associate to the peaks. So we could go in a little bit greater detail, and in this case, we're just pulling variants that co-localize with annotated Ts. You'll see we mostly recover that middle peak again. And then we can do the opposite and sort of focus on SNPs that are not in Ts and SVs that are not in Ts, and we recover most of the signal in that case. So just to summarize what I've shown you today, in this system, there's few genomic regions that differentiate species or associate to coloration differences. Most of the differentiated SNPs are in non-coding regions, and we have actually few species-specific variants at these peaks. What's most common is that species share variants in unique combinations across the different regions. And finally, our differentiated markers are mostly SNPs and smaller indels, both within and outside of Ts, but larger SVs don't seem to associate to species differences. And so I will leave my acknowledgements there, thank the team, funding sources, and I may have like